this lecture, we'll discuss about how acetylcholine is made in the neuron and then how does it work on the membrane here, on the postsynaptic membrane. In the same way, we'll discuss about norepinephrine or epinephrine, dopamine, the whole family. So when it comes to the formation of acetylcholine, we said that cholinergic neurons are producing acetylcholine, and then it is released and then it works either on the nicotinic receptor or muscarinic receptor. We'll see that this is, the neuron here we call the presynaptic. This is the synapse. Here would be the synapse, and this will be the presynaptic membrane, and this is the postsynaptic membrane. Now we know that acetylcholine is made here in this neuron here. Now we know that choline that is uptaken back and acetyl CoA that's made inside, both of them when they combine together by choline acetyltransferase, this is the enzyme, and then it's going to make acetylcholine, and then it's taken inside a vesicle, right? It's a vesicle, and to protect it from outside, then it will be stored in the vesicles. Now, once we have an action potential coming from the body towards the axon terminal, and it become action potential is coming here, when the depolarization wave reaches here, what's going to happen is that it's going to open the calcium channel. Calcium is going to influx from outside into the cell, and this is going to cause exocytosis, which means release acetylcholine, burst this vesicle, and it will come into the synapse. Once it comes into synapse, it will go into postsynaptic membrane. On the postsynaptic membrane, there will be acetylcholine receptor. We said that the acetylcholine has either nicotinic receptor or muscarinic receptor. Once it works on the receptor here, the function that it has to do has been finished, then there's an enzyme called acetylcholinesterase. This is the enzyme. This enzyme is going to degrade acetylcholine into two steps, into acetate and choline. We don't need acetate anymore because our body can produce it, and then it will be reuptaken. Now the speciality is that the whole stuff will not be reuptaken, only the choline will be reuptaken, and you could see the choline is reuptaken. When it's reuptaken, again body produces acetyl CoA, it binds with choline, and then it will be taken back. So this is an important cycle. So we should remember, important is this enzyme, choline acetyltransferase, storage here, action potential coming here. Once action potential comes here, and then it will be released. Once it's released, works on the membrane, function is finished, then there's an enzyme, the enzyme degrades it. We don't need acetate anymore, choline will be reuptaken back. This is important. Now we also know, you could see that we have, if you could see there's some pharmacological effects here. What are the pharmacological effects? Do we know botulinum toxin? Botulinum toxin, it's released by a bacteria. Now that's a deadly bacteria because it's releasing this exotoxin called botulinum toxin, and we use also Botox a lot in cosmetics. Now what does it do? Why does it make the muscle relaxed? Because it stops the release of acetylcholine here. So this is an inhibitor here. That's why at the neuromuscular junction you have nothing. You don't get anything. That's why you don't have muscle excitation. That's a Botox or botulinism. We use it in a very small amount because it can really kill you. Second thing is, we also have, if we want to increase acetylcholine in the synapse, thus increasing the activity of acetylcholine, we have drug called acetylcholinesterase inhibitor. If we have acetylcholinesterase inhibitor, then what will happen? The effect of acetylcholine will increase. We'll study this in another lecture. Now, let's compare this, cholinergic or acetylcholine formation and, release and effect, and then degradation. Let's see it with another, important family of neurotransmitter like noradrenergic system, such as epinephrine, norepinephrine, dopamine. Even we have for we have for serotonin. Here we know that this is mostly released by a sympathetic nervous system. We have an exon, we have an amino acid tyrosine, tyrosine is changed into dopamine, dopa is changed into dopamine, and then it can go ahead and it can make epinephrine and norepinephrine. Now, we studied before that inside the nervous system we'll produce a lot of norepinephrine, while in the suprarenal medulla we have a lot of epinephrine. We studied. This is stored here, and this is a very important transporter. We MAT. This transporter takes it inside. If you don't take it into the into this vesicle, it will be degraded by enzymes. Here we have two important enzymes called COMT. They're COMT and MAO enzyme, right? They are very important enzyme to degrade them. One is outside, and one is the other one is inside the mitochondria. And then in the same manner that action potential will come here, action potential comes here, and it as it reaches at the terminal, it's going to open the calcium channel. Calcium comes inside. Once it comes inside from outside, this is going to cause the exocytosis, and it can come into the synapse. Once it comes into the synapse, right, diffusion, then what's going to happen? Then it's going to work on alpha receptor or beta receptor, as we discussed it. As the effect on the postsynaptic membrane is finished, what would happen? This epinephrine or norepinephrine cannot be degraded. It will not be degraded. It will be reuptaken. You could see this is the norepinephrine transporter. This is an important transporter, the reuptake. So inside the synapse, we don't degrade them and then, like we did there, we don't degrade them, we reuptake them. So it's slightly different. We reuptake everything back. Once we take everything back, and then what will happen? It will be taken back. Once taken back, either it will be degraded like inside the terminal, either it will be degraded by COMT or MAO, or will be taken back into the synapse. Here you can understand, you could see different drugs. Do we remember, like amphetamine or ephedrine? Amphetamine, we know that this is a CNS stimulant. What does it do in the central nervous system? It increases, the ephedrine is in the same way, it increases the release of epinephrine and norepinephrine in the central nervous system. We have like other drugs, which is like tricyclic antidepressant, especially tricyclic antidepressant, and we know cocaine. These are going to stop the reuptake of epinephrine and norepinephrine, and when they stop the reuptake of epinephrine and norepinephrine in the synapse, it's going to increase, and it's going to have an effect. And in central nervous system, we know that it's used for antidepressant. We also have alpha 1, alpha 2 receptor on this presynaptic membrane. What is the effect of alpha 2 receptor? It's working as a negative feedback. What does it mean? If we release, let's say I'm really releasing a lot of epinephrine and norepinephrine. Now, how would I know that there's a lot of epinephrine and norepinephrine? What will it do? The same, epinephrine and norepinephrine is going to bind here, and it has an inhibitory effect, and it's going to stop the release. Thank you very much. I hope you enjoyed the lecture. We'll see you in the next lecture. Thanks a lot.